Okay, good morning, everyone, or as Jan suggested, just a good, depending on what time zone you are in the world. My name is Ryan Reisinger. I'm the incoming Deputy Chief Officer of the expert group on birds and marine mammals, and I'll be leading this first session today. Try to limit the talks to about 12 minutes. We only have 15 minutes per slot, and we need just a minute or two for changing between the speakers and perhaps a question or two. I ask all the speakers to please try and stick to that 12-minute limit. Don't have too much housekeeping. If you have questions for any of the speakers, please pop them into the chat box and I'll try and monitor those and put questions to the speakers. If we do have time at the end of the session, then we can revisit any questions we didn't have time for after the talks themselves. I think that's all from me. So without any further ado, we'll start with our first speaker, Sarah LaRue.
Thank you, Ryan. Should I start? Yes. Good morning, good afternoon, good night, everybody. I don't know where you are on this planet. So today I will present you some work I've been doing in collaboration with Michel LaRue and Stéphanie Genevrier, mainly at the University of Minnesota and Woods Hole Oceanographic Institution. And I'm now finalizing this work. So today I will speak about where to live and the role of fast ice characteristic in shaping the presence of emperor penguin colonies around Antarctica. So as you may know,
Can you share your screen, Sarah?
Sorry?
Can you share your screen?
Oh, the screen is not shared. They remove it?
Yep.
Okay. That would be useful.
Sorry to interrupt you.
No. I was presenting earlier. Oops.
That's the green button at the bottom of your. It's not
yes. Yeah, but my diaporama is presenter. Okay. And now I can. I can't reach it. Should be fine now. Do you see it well?
Oh, good.
Okay, so let me know if there is more problems. Okay, so we're talking about emperor penguin still. And I go there. Here we go. So we know that what we are observing now in terms of carbon dioxide in the atmosphere is more than it has ever been. And it's well above anything we have ever experienced as humans, and will continue to rise in the future. And this figure shows the greenhouse gas emission projected under a business as usual scenario. Will go well above, and we continue its current course with dramatic impacts on population, ecosystems, biodiversity. And today I'm talking about emperor penguins. Indeed, these species are sea ice obligate species. That means that they are always found near the sea ice, and they need the sea ice in Antarctica as a platform for reproduction and for also resting. And they are breeding during the winter time, and they are doing some journey at sea to feed and raise their chicks. And the duration of their journey depends on the extent of the surrounding sea ice cover, but also the stability of this sea ice cover, and also the proximity to water openings from the colony. to be able to feed and return to the colony regularly. Thus, both the stable sea ice and water openings are essential for adults to maintain their body conditions, their survival, but also to successfully raise their chicks. But the problem is that in Antarctica, here are the sea ice trends from 1982 to 2017 from the IPCC report. We are experiencing some very contrasting regional trends in terms of sea ice that are different where we are in Antarctica, and that will affect emperor penguin population in different ways. And also there are a strong enigma in terms of the Antarctic sea ice trend, because since the satellite era started, since 1979, we observe kind of a positive trend, but we observe also some abrupt change in the sea ice. For example, here in a red rectangle, where we can see some dramatic decrease that we are struggling to explain, and even the model simulation are not successfully representing the observation. And so we don't know how the sea ice change will affect the population, and there are a lot of uncertainties around this. There have been different works that have been trying to project the emperor penguin population using model simulation with sea ice and population model. And if we are going to a reduction of sea ice and an increase in the temperature in the atmosphere, this paper from colleagues and especially Stephanie and Michel are showing, this paper is showing that there will be a decrease of 98% of the colony if we have an increase of 2.6 degrees Celsius in temperature. And what is interesting here is that you can see on this part of the graph that there are strong difference between the regions in terms of how they answer to different change in temperature, and there are regional difference. And from this work, we were wondering what are exactly the drivers of emperor penguin presence around Antarctica? Can we find some, and if any, how do they drive the population of emperor penguin around Antarctica? So I will talk a lot about fast ice. So here it's a satellite map of different type of sea ice. Here you can see the East Antarctic ice sheet that is attached to the continent. And attached to this ice sheet, we have fast ice, which is a region of sea ice where you don't have too much leads and cracks. It's very uniform. And then you have the pack ice region and the marginalized zone. And the analysis I will present today will mostly talk about the dynamic of this fast ice, because it's where the emperor penguin spend their whole time breeding. So for this analysis, we considered 13 variables to try to explain the presence of emperor penguin around Antarctica. And first of all, some fast ice variable that have been studied from the work of Alexander Fraser. So from 2010 to 2018, we have fast ice variables at 1 kilometer resolution that are the persistence of fast ice, which means like per pixels over the 18 years, how much the fast ice is persistent within the pixel, the fast ice strain, volatility, which is an index of how much the fast ice breaks and forms, the fast ice extent, the magnitude of the seasonal cycle of this fast ice, so the difference between the fast ice minimum and maximum, and how does it change from one year to another. And also the timing, which means is the fast ice minimum or maximum early or late. We also consider some topographic variables and biological factors such as intra and interspecific composition, which mean like mostly distance to other species such as weather seals or Adelie penguins around the colony of emperor penguin. The main problem we have is that we have 55 colonies, and Then based on a grid of 5 kilometers, we then have 177 pixels of presence, but we have 58,580 pixels of absence within the fast ice area. And so this was a problem, so we can't really use regression analysis because the difference between the presence and absence is so important. So we decided to, in our case, use a principal component analysis. So we ran a principal component analysis on the presence data. We also observed that the presence was not linked to a single variable and was not also very strongly differing from the absence when we did some just distribution plot. So the idea of running a PCI will allow to kind of characterize a combination of variables that represent the presence of the penguin. And then based on this principal component analysis, we then projected all the absence pixels conditionally to the structure of the presence in the PCA space. And the idea of doing that allow us to then increase the difference between the presence and the absence. For example, environmental condition of an absence value far from environmental condition from a presence value will be even greater within this analysis. And then based on the presence and absence described in the PCA space, we ran a clustering analysis to define some type of habitat. So here is just an example. I couldn't represent the 13 variables for this talk, but just an example of the fast ice volatility with the fast ice persistence at the bottom. The gray dots, the thin gray dots represent the absence and the plain white dot represent the presence. And as you can see, it's very interesting. We have a bell-shaped curve in terms between the volatility and the persistence, but we can't clearly see some patterns between the absence and the presence. So this is the interest of running such a type of analysis. So I will go quickly over that, but basically we ran this principal component analysis, and it was interesting because the variables were orthogonals, and so it means that they are totally independent from each other, depending on the dimension, and means they bring different percentage of variability. And we can see here that the most dominant and contributing variables were the fast ice persistence, its magnitude of the annual cycle, but also some bathymetric features such as the topographic slope. I will not detail the analysis for 4 components instead of 2, but we find some similar variables, but we increase the number of contributing variables. Then we ran the model-based clustering, and when you run this model-based clustering on the presence data from the PCA, you obtain different classes for each colony. So here are the different emperor penguin colony, and we obtain 5 classes. And what is interesting is that we didn't take into account the geographic structure in the analysis, but in a PCA analysis. But then when we project the result on this map, we observe a geographic structure, which is very interesting. So we can characterize using these 13 variables some different habitats that make sense in terms of the geography. So we found that we have a region characterized by class 1, the eastern part of the Weddell Sea. Then we have a second region characterized mostly by the class 5, but we have, interestingly, 3 colonies with very specific habitats because they are the only one to have the class 3 among all the Antarctic continent. And then we have the region around the Ross Sea that is mostly characterized by the second class. And then we have the rest of this region that is a mix of different type of habitat, interestingly. And then if you apply this model-based clustering on the absence data, and based on 80% certainty of belonging to certain class, we can also predict around Antarctica where this kind of class of habitat could be, where the colonies are not. So here is a map representing the different type of habitat, and we found a similar pattern. And then based on this habitat, we can then look at different type of variables for each class. So here it's just an example for 6 variables, and you have like for each variable, each class that I represented in terms of habitat. And then each box plot, as you can see, the lighter color is the presence and the darker color is the absence. And what is interesting here is that we didn't find some very strong difference between the absence and presence pixels. However, we found some significant difference between the type of habitat and the type of cluster we identified. So I won't go into a lot of detail, but I will just show for the first group. So for example, this group in red, we can see that we have medium, intermediate, fast ice persistence and volatility, relatively low magnitude of the annual cycle, low fast ice extent. And this will differ from, for example, this group in green, where we can see like strong persistence, intermediate volatility, important magnitude of the annual cycle, still low fast ice, and the trend neutral. So this is interesting to look at what are the different variables representing the different habitats. So I will switch. So the next steps that I won't present today is, we run also this analysis, is the population dynamic linked with the different habitat identified? Because from recent work, we obtained some emperor penguin colony trends over 10 years around Antarctica using satellite data and some Bayesian modeling. And then now we can also link this with the habitat. So I will conclude this by saying that this study is interesting in a way that it shows that there are different types of habitats among emperor penguin colonies. So all emperor penguin colonies don't have the same type of habitat, and we can find some strong difference between the different habitats for certain variables. It was also very interesting to see that within fast ice, so within like the habitat that is available and possible for the emperor penguin to breed, there is no strong difference between the presence and absence area, but there is strong difference between the type of cluster. And I think it will be very important now in the near future to also consider pre-availability and the importance of functional ecology and predator-prey relationships that might be a strong driver as well of where the penguins are and where they can actually feed and forage successfully. And to finish, this is a map of the habitat suitability. So it's a probability to be in the best habitat based on the relation identified with the variables. You can then predict where it is suitable for them to be. And if our knowledge in terms of the observation we will get in terms of fast ice and different dynamic variables increase in the future, it's a good tool then to improve, to then predict and observe where the habitat becomes suitable or unsuitable. Thank you for your attention, and thank you to all my colleagues that helped me to do this research.
Thank you very much for that presentation, Sarah. Just remind you that you can put your questions in the Q&A box. We don't have time for any questions now, so we're just going to move straight on to our next speaker, Nadia Kepper.
See my screen? Okay, so thank you for the introduction and thank you for the nice talk on the penguins. I'll now present you something about Wilson storm petrels in Antarctica and the consequences of heterothermy on their physiology and growth. The Wilson storm petrel is the smallest endotherm
that is living in Antarctica. And while the birds spend most of their time in the oceans, they will come to ice-free areas of the Antarctic and subantarctic for breeding each year. They are breeding in scree slopes, as you can see on the left, and put a single egg underneath big rocks, where they incubate it until their chick hatches. For the first couple of days, the parents will stay with the chicks, well, with their chick. But after already about 5 days, the parents will leave to forage during the day and only come back during the night to warm the chick and also to feed it. So the chicks are developing the capability of maintaining their stable body temperature very early in their life. But about the same time, they're developing also the capability of decreasing the body temperature again and the metabolic rate to save energy when they don't have enough energy to maintain their body temperature. So if they don't receive enough food. And this happens regularly because the parents are often staying away for more than 1 day for foraging trips. But also, as you can see on the right, there are regularly snowstorms blocking the nests and the parents cannot enter. During these times, we could show that the chicks can survive these elongated periods without food by decreasing their body temperature. But we are interested in what consequences this behavior may have on their development. In particular, we want to know whether it has an impact on their growth, and we are looking at this for the tarsus, so part of the chick's leg, the impact on senescence and on immune system. When I talk about heterothermic animals, it means that it's nestlings that have body temperatures below 36 degrees, and the normothermic body temperature, so the normal body temperature is usually around 39 degrees. The heterothermic is not an on-off thing, so it's a rather on a scale, but we could show in earlier studies that only about 10% of all measurements were below 36 degrees, and therefore we decided to keep this as a threshold for further analysis. We conducted the study on King George Island, which is an island north of the Antarctic Peninsula on the Kalini Station. And we there had one colony of Wilson storm petrels that were monitored daily. So when the chicks were old enough that the parents were leaving during the day, we went there every day to measure the body temperature. We measured the tarsus length, so this part of the leg of the chick, every 3 days, and we sampled blood from the chicks every 7 days. The blood was centrifuged and the plasma was used for immunoassays and the red blood cells for telomere length discrimination. To see whether we can see an impact of body temperature on the tarsus growth, we first got an overall growth curve from according to age, the tarsus growth from several seasons of the nestlings. And then we looked at one particularly day and compared the last tarsus measurement with the next tarsus measurement. Here you see the last measurement in green and the next measurement in blue. And we wanted to know whether, depending on the body temperature at the red line, there will be a change, so that before the last measurement would be, for example, above the curve, and then after heterothermy, the next measurement might be below the curve. For senescence, we looked at telomeres because telomeres are important to protect the DNA, and their shortening is associated with cell death. So if the telomeres get too short, the coding part of the DNA will be damaged and therefore the cell will die. Hence, maintaining the telomeres is costly, and shorter telomeres predict usually lower survival probabilities. For the immune functions, we looked at both the innate and the adaptive immunity. The innate immunity is the immunity that the nestlings are already born with, and we conducted a bacterial killing assay to test this on the blood plasma. So how much or what percentage of bacterial the plasma was able to kill. And the adaptive immunity is considered to be rather costly. And here we looked at the antibody levels of the IgYs. For the tarsus growth, we first wanted to know whether we already see a difference between the before and after measurement when we have homotherm animals, so with a normal body temperature. And we could see that there's no difference. So you see the green before curve and the blue after curve, and there's no statistical difference for both curves. But if we now look at days when the chicks were using heterothermy, so they had body temperature below 36 degrees, we can see that the green before curve is still pretty much well around the zero from deviation to the general growth curve. So this means that an equal number of chicks is above the growth curve or below the growth curve. But when we compare this now to the after measurements, we see that we have a shift towards smaller tarsus length compared to the general growth curve, which indicates that the tarsus growth slowed down during the heterothermic event. When we looked at the telomeres, we could see that, we looked at telomere shortening. So this means we looked at the samples from the same individual at 2 points in time, usually about 20 days or at least 20 days difference between both measurements, and then compared how much the telomeres would shrink. As this is also associated with age, we looked at shortenings per day to exclude the age from this analysis. And then we compared it with the mean body temperatures during the same interval. And we could see that the shortenings were stronger when the mean body temperature was lower. So indicating that chicks that were using heterothermy more often had faster telomere shortening and therefore a higher chance of cell death and senescence. When we looked at the immune system, we could not see an effect of the body temperature on the innate immunity. So body temperature didn't seem to affect how well the plasma was able to kill the bacteria in the assay. And we neither saw a significant effect of the adaptive immunity, but levels of IgYs had a tendency to be a little higher when the mean body temperature until the measurement was higher on average. However, the sample size is not huge, and adaptive immunity is also known to be correlated to age and sex, for example. So we kind of had to include these factors in the analysis, which makes obviously the need for more data higher. And so would be interesting to see with a bigger data set whether we can see really an effect of heterothermy on the adaptive immunity or not. So to sum it up, heterothermy is influencing tarsus growth negatively. The senescence will increase, so that's also not good for the nestlings. And the immune system may not be that strongly affected, at least what we can tell from our data. Coming back to the title and the topic of the whole conference, Wilson storm petrels in the changing world. The changing world here is affected because for the Antarctic Peninsula, it's predicted that snowstorms will increase with the climate change. And this is also increasing the chances that the nests will be blocked by snow and the parents cannot access the nest, and hence the nestlings will need to use heterothermy more often to survive at all. And while this enables them to survive, which is of course great and amazing, they may fledge at a less developed state than they could with less snowstorms and less use of heterothermy. Thank you for your attention, and if we have time, I'm happy to answer questions.
Thank you very much for your talk, Nadia. If there are any questions, could you please put those in the Q&A box or raise your hand if you'd like to ask that question live. We have a minute or so for that. Okay, we can save any of those questions which come up for a bit later. In the meantime, I'm going to ask our next speaker to get ready. That will be Lynn Asfestad, speaking about passive acoustic monitoring at the South Orkney Island.
Yep. Can you hear me all right?
Yeah, we can hear you, Lynn.
All right. Then that should be my screen shared there.
Yeah, we can see that.
Yeah, all right. Perfect. So then we will move to a bit of a bigger animal. So I will talk a bit about the marine mammals and their acoustic presence around the South Orkney Islands. So just a quick introduction first. So the passive acoustic monitoring is a very good non-invasive method that can be used to gain high resolution information about the marine mammals' acoustic presence because they are so vocal. And so multi-species PAM studies can help increase our current understanding of the temporal change in species diversity and assemblage, and that is especially useful in remote areas such as the Southern Ocean, where regions are inaccessible for a very large part of the year. And so South Orkney Islands, which are located in the Scotia Sea, has been recognized as a hotspot for Antarctic krill, which is the main food source for a lot of the migrating marine mammals and residential. And so the northern side of these islands is among one of the most important commercial krill fishing grounds. And then such hotspots will naturally attract both residential predators, which then will be the Antarctic seals, for example, and then also migratory predators, which will be more of the large baleen whales. And so due to that assumed high krill concentration in this region, it is essential for us now to try to gather more information about the major krill eaters that roam in this region. And not only to implement that information into the management of the fishery, but also in light of that ongoing climate change, and especially because of the Antarctic Peninsula and Scotia Sea region being one of the most fastest warming areas on Earth, and that rare weather events such as ENSO are likely to increase in both frequency and intensity in the future. It is crucial to increase our understanding and knowledge about how this may affect their distribution and abundance. And so the data we used in this study was collected through an oral that was attached to a mooring anchored to the seafloor in the Coronation Trout, which is northwest of the biggest island of the South Orkneys. And despite this area being such an important krill fishing ground, information and knowledge about the marine mammals and their community structure here represent a knowledge gap. So we do know that there are whales and seals here, but information about the extent and which species and when they are actually located here is still pretty scarce. And so in this study, we had acoustic data from 2 years, from 2016 and 2017. And 2016 was characterized as one of the strongest El Nino events on record. And this gave the opportunity to not only look into let's say, the basic species acoustic presence, but also how kind of such weather anomalies affect the acoustic phenology of the marine mammals. And so in 2016, we had data from February until August, and then we had 12 minutes of recording per hour. And in 2017, we had only 8 minutes of recording per hour, but then we also got 2 months more of recording, so until October. Yeah. And so basically what I did was that I visualized all of the roughly 10,000 hours of recordings as spectrograms, as you see here, with the frequency and time. And then I skimmed through these for vocalizations, basically. And I primarily used Ishmael bioacoustics, which you see here, with the pretty wide frequency range from 0 until 8 kilohertz, to kind of increase the possibility to get all of the calls for the potential species that may be there. And I also used Python Audio Spectrogram Explorer to look in a more narrow frequency range, only the bottom part, to annotate Antarctic blue whale and fin whale calls, as these are very low frequency and were kind of hard to see or differentiate between in Ishmael. And then just quickly about the environmental variables that I used. So I used spatially restricted environmental covariates. So I used SST, SSH, sea ice, and bathymetry. And then they were spatially restricted within the audible area of the oral. So here's the South Orkney where the oral is placed, and here is the SST for 1 day. So those covariates I put into the CCA, where I looked at large scale patterns in the community structure for all of the species, and then I used them in GAMS to look at the impact of each of the covariates on the species that I had most data on. And so when I looked at the patterns in the sea surface temperature and sea ice during the 2 years of data that I had, I observed some clear interannual variations. And so firstly, there was a clear drop in the 2016 spring sea ice. And that is a probable result of that increased SST in the springtime, which was much higher in 2016. And these kind of weather anomalies continue to affect the summer and fall season into 2017, where you see that the temperature is much higher compared to the previous years. And then this increase in temperature then continued to affect the sea ice in the 2017 winter, which shows almost a month lag or a delay in the sea ice formation. And all of this is a probable result of that ongoing El Nino event. And as krill is such kind of affiliated with low temperatures and ideal sea ice conditions, such anomalies will or can presumably strongly affect their survival and distribution. And that in turn will affect where the marine mammals are, because they are here to eat. So based on those 16 months of acoustic data that I had, we found 5 baleen whale species, 2 pinniped species, and then odontocetes, which were not separated by species. And despite being little change in the level of species diversity over time, there was an almost complete shift in the gill composition related to season. So we had the ice avoiding species, the southern right whale, fin whale, and humpback whale, which were more closely associated with the higher temperatures and low sea ice concentration. And then we have the ice-affiliated species, the odontocetes, minke whales, and the 2 pinnipeds, leopard seal and crab-eater seals, which then showed association with higher sea ice concentrations and lower temperatures. And then we have the blue whales, which didn't show any clear association with any of the variables. And I will come back to that in a minute. So that high degree of seasonality was further shown by interannual variation in the species acoustic phenology. So here is a time series of the acoustic presence of the blue whales. So they were observed year round and hence no clear correlation with the environmental variables. And such year-round presence indicate a more partial migration within the population may, where some individuals, such as immature animals or females, stay back on the feeding grounds to conserve energy, for example. But for the blue whales here, it is important to note that you can see the sec call of the blue whales here, which I annotated. But for the most part, I only saw this A unit, the top part, and that is the strongest part of the call, and that can propagate for a longer distance. So what is most likely, I think, is that these animals are further away or moving further away from this area towards or during winter time, because these complete sec calls were only spotted in the beginning. And then we have the ice avoiding species, humpback, fin, and southern right whale. All of these three species presented a very similar pattern, where the cessation of all acoustic presence happened before the main ice formation or austral winter. And these time series kind of reflect their common dissociations with the sea ice, and was further shown by their 1 month extension in acoustic presence during 2017, when the ice formation was delayed. So they stayed 1 month longer in that year. And we also saw an additional peak in 2016, which we didn't see in 17. And this may reflect that there were more unsuitable conditions, or simply that it wasn't enough food, which may have led the animals to relocate elsewhere before they returned at a later time. And though we can't say anything about the arrival time of these animals because we sadly lack the summer data, we do get an idea of their departure time back north to breeding grounds as the winter and sea ice progress and reduce their kind of accessible area and prey availability, and that kind of pushes the animals out of that the oral's audible range. Yeah. And then lastly, we have the ice-affiliated species. So we have the minke whale and the two pinnipeds, crab-eater seal, leopard seals. And these kind of presented a contrasting pattern to those that I just talked about. So they were only heard during winter or springtime when there were periods of heavy ice cover. And so the minke whales are well adapted to live within the pack ice due to their size and body composition, in contrast to the larger baleen whales. And the two pinnipeds are ice breeding and rely on the sea ice during breeding season. They rely on it for feeding, resting, and molting and popping. And it was for the ice-affiliated species that we kind of saw the clearest interannual difference in acoustic phonology. So the minke whale acoustic presence were noticeably higher in 2016, while the two pinnipeds were virtually absent during that same year. It was only leopard seal that appeared in 1 or 2 files. So while the reduced presence of minke whales in 2017 may be a result of a more interspecific competition with the ice-avoiding species, which extended their stay that year, or simply them relocating due to non-ideal conditions. This is all assumption, but it's a very interesting subject that is really worth looking into and continue looking at. And for the seals, their disappearance or non-acoustic presence in 16 shows how much they rely on that sea ice that suddenly disappeared that year. And all of these results give us a good indication of how the different impact of changing environmental conditions. But I only have 2 years of data, so I have to be careful how many conclusions you draw. But anyways, this was kind of the first study looking into the marine mammal community structure at South Orkneys through passive acoustic monitoring. And it clearly showed that this is an important feeding ground and year-round habitat for a range of marine mammals. And as the fishery and these animals utilize the same resource, there needs to be some sort of resource partitioning and knowledge about the marine mammals regarding how much food is necessary to support their populations, and when they actually feed in this region is crucial for the future management and conservation. And because the environment keep changing, we need to understand how these animals respond to these changes, as we have seen here that they can relatively quickly alter their habitat use. And so for this particular study and continuing work, getting year-round PAM data, and especially that summer data, is essential for getting that full picture of their movement in and out of this area. And it will be cool to kind of try to link this directly to hands-on on krill data. So yeah, that was it for me. Thank you very much.
Thank you very much for that talk, Lynn. We can't seem to find our next speaker, unfortunately, so we will have about 10 to 15 minutes for
I think
questions.
I saw him in the, it is my supervisor, that's the next. I think I saw him in the attendees list and not the panelists, but
Okay. Yeah, Reisinger raised the hand. So yeah,
that's it.
Okay, I've just
Right, sorry about that. I'm not sure why that's my Google account and not the work account.
That's
Sorry
all right.
about that.
I'm glad you're here. So our next speaker will be Sebastian Menz. Fortunately, Sebastian, I hope you are able to share your screen. Let me know if you're not.
Not yet, not yet.
Okay, let's try and sort that out. Okay, I'm just going to promote you to panelist. Okay, that should work now.
The master view, nice.
There we go, perfect.
And let's go. All right, all good?
All good, thanks.
Nice, okay. Yeah, thanks for the opportunity to present some of our research there. I joined this Norwegian krill research around 2 years ago. Still postdocing there, and like to use this opportunity to show you what we're looking at, what we're working at. And yeah, I guess we haven't been that present with the South Orkney research at SCAR that much, but I think that's about to change. Lynn gave a really nice introduction on the South Orkneys, and I'm just going to add a tiny bit on this. And the part I'm going to be talking about here are our visual surveys and our line transect surveys that we are basically doing since 2011, missing the Corona years, unfortunately. And the area we are looking at is the shelf north of the islands, but also south of the islands here. Usually performing 5 line transects, and maybe some more. Some years we have some concentrated transects north of the islands as well. And as Lynn said, the reason for this is an increased presence of krill fishing vessels in that area. So there was a nice paper by Bettina Meyer that basically just plotted the catch data that is reported to this Kamlar Conservation Society, every year, every season. And the last 10 years, we could really see a clear rise in the South Orkneys, and especially the shelf and the canyons and the shelf north of the islands as a hotspot for krill catch landings in the summer and spring month, whereas the winter season is more South Georgia and Bransfield Strait. So that's one of the reasons why I'm the Institute of Marine Research here in Norway, and the Polar Institute is really looking into that, because Norway is also one of the largest fisheries nations, and we want to make sure that things are sustainable there. And the vessels we use are either fishing supply or actually fishing vessels itself. And one year we actually got lucky and used our big icebreaker. And an example for such a survey, the results is what we do. We get down there end of January, start of February, and we want to find out what's the Antarctic krill abundance using acoustics, like the echo sounders that are usually calibrated in one of the base, either one of those calm areas south of the islands. And then we're also doing catches, so using trawl stations and CTD stations in intervals. Unfortunately, this year was plagued by a lot of technical problems, so we don't really have catch data, but we have really nice echo sounder data. Alongside that, there's marine mammal observations with a dedicated observer during the times of good visibility, and we're also deploying those moorings and have some drones that I'm going to be talking about in the end. Here's the data from this year. I'm probably going to shove this away. There we go. So here's the data from this year. There's just a spatial interpolation of the krill density, where the green under the yellow indicates areas of a lot of krill swarm presence, and the black lines are the acoustic echo sounder data. The red dots here are the fin whale sightings that we made, and you can see that there's quite a lot of them in the north this year, and we definitely saw quite a few, and some in the south. And the krill actually really nicely lined up along the shelf line north of the islands. They're indicating that this area really is quite interesting. And one of the reasons why both the whales and the fishermen are there is actually a supply of krill coming supposedly from the Weddell Sea. So there's a shelf current that flows from the east towards the west, supplied by the Weddell Sea, but there's also the Antarctic circumpolar current in the north, and it's a very dynamic environment. So the currents there's tons of eddies. It's really quite interesting. So there's a high water mass exchange on this shelf slope going on here, whereas the shelf in the south is a lot more it's more stagnant water masses. So what we do with those marine mammal sightings data is we crunch them using distance sampling. So the observer usually looks at the single forward quarter, like either the left or the right side, or starboard, and what's the English name? Whatever, you know what I'm talking about. So one of the 90-degree quarters, we are logging the location, the species, the group sizes, the angles, distance, and visibility, and sea state. And then for each survey, we're gonna look at detection function, because the vessels and observers change between the years. So we, from these detection functions, we can then get our detection probability, and then get for each transect a density of the animals observed, so individuals per square kilometer in that instance here. So here's an example for this year's survey then, where for various reasons, not all transects are covered equally well with weather and storms and waves and whatnot and operations, but on some of those sections here we get useful transect data. And those are really interesting because they're really fairly high, right? There's a lot of empty transects where we didn't see much, especially in the south. But here in the north, holy schmoly, that's a lot of fin whales. That's some really large numbers. So that was pretty cool to see. And if we crunch together all data from all years, we can have a more of a look at, is this spatial distribution here a single event, or is there some more trends? That's what I'm showing here, where in this case, I'm comparing the distribution of humpback whale sightings to the fin whale sightings here on the right side. And the first thing that meets the eye is that these are really quite separated. Humpback whales are mainly observed on the shelf, or a little bit along the shelf edge here, whereas, and also in far less numbers. So I forgot to mention that the #1 species we observe are the fin whales. Then there's humpback whales in some years, and then some minke whales and southern right whales. But definitely those two here are the number one and number 2 in terms of abundance that you observe at sea. And the fin whales are really associated with these offshore areas here, where there's a lot of dynamic oceanography, and also quite some krill swarms that we encounter. And we can look at this over time actually now, because we have these 11 years and we have each survey for each year. And then it gets quite interesting because that's some pretty cool data, these time series of marine mammal abundance. And let's start a bit up here on the upper right side with the fin whales. We have our time series over these up to 10 years, and I split the analysis here into the northern section, north of the island, and the southern section. And first thing we'll see is, wow, there's a lot of variability between the years. Sometimes we observe almost no fin whales, and especially in the south of the area on the shelf, we observe quite a few fin whales, except one large group that kind of skews the statistics here a bit up. But in general, there's a lot more fin whales in the north, which makes sense as well, because that's where all the krill is as well, mainly. And then also to note is that these are really quite high numbers. That's some pretty high fin whale densities there, and that is yeah. I was also like, oh, wow, we have to be careful. This is real, but I checked all the data. It's like yeah, we are. It looks very solid, I would say. We are careful in our analysis. And then earlier this year, there was another paper by Helena Hare on the feeding aggregations near the Antarctic Peninsula. And they're actually showing quite similar values here, around 0.2 individuals per square meter. And that is very comparable to what we are seeing here. So this really hints towards a successful rebound of the fin whale population in the Scotia Sea, and they're really one of the major krill predator populations that has bounced back. If we then look onto the humpback whales on the other side, there we see quite a different pattern there, and also a far smaller magnitude. So there's far less humpback whales in the South Orkneys than fin whales, at least during the January-February time when we're there. And here you can really see some interesting pattern that looks different, and we'll come to that in a second. The next species that we observed there at sea are Antarctic fur seals, which are they're not a pelagic species. They're breeding on the South Orkneys, but their at sea distribution here shows some interesting peaks there. Now, what could be the reason for these patterns? They're all snapshots in time, right? So each survey is just like a 1 week snapshot. So it's hard to it's a bit hard to compare them, but we can at least give it a try and see how it compares. So the next thing we want to, of course, look at what's their prey doing, their main prey here, the Antarctic krill. And the time series in Antarctic krill abundance shows that, again, the northern section here has the most of the krill, and the southern part sporadically a little bit less. Or in general less, but sometimes it changes. And then there's one year that really stands out. That's really interesting, and that's the one that Lynn already talked to you about, and that's 2016, where we have this ENSO year, which has the lowest krill abundance that we recorded, has the highest humpback whale abundance, and also one of the lowest fin whale abundance there. And I'll just show you a figure again here, another figure where I'm actually plotting this El Nino index. There's two ways of calculating strength of El Nino. They're more or less the same here. And you can see 2016 here. Oh, wow, that is a really strong peak. That's a very strong El Nino event, which coincides with the strongest humpback, highest humpback abundance and among the lowest number of fin whales. In this case here, this is average now for the entire study area. And when you put it into a correlation analysis, it's not that many the data points, but it does show a significant correlation. So that was pretty cool to see, actually, and goes well with other passive acoustic studies in the Weddell Sea, which also finds that really humpbacks shift their habitat quite a lot during these ENSO years. When we look at the connection between the Antarctic krill and the ENSO, we also find a little bit less stable and a little bit less strong correlation, but it's surprising that those snapshots in time, because these surveys are just such single snapshot, and the krill patches, if you ever looked at krill data, it's terribly non-linear. There's these small patches here and there, and then there's a lot of nothing, and then there's a lot of patches again. So all you get are these snapshots of this very dynamic dynamically distributed population. But still, yeah, through all that noise, we actually still get these cool climatic connections. The things are looking a little different for Antarctic fur seals, where we didn't find a strong connection to ENSO, but we found a strong connection between their presence at sea and the sea ice edge latitude here. So when the sea ice, when there were strong sea ice years, so in this case, the blue line here shows how far north does the sea ice edge reach. And when our sea ice edge is very close to the islands, or partially even north of the islands, then we observed a large number of fur seals, which would make sense, I guess. The things, yeah, those findings here, we're in a moment working on publishing those, and we will continue these surveys for as long as we are getting funding, I guess, which yeah, looks quite okay so far. But what we want to know is these surveys are all snapshots, and these are noisy, and we want to know what's going on between the surveys, what's going on in winter, spring, fall, all those times we can't be there. And there have been two approaches to look at this so far. One of them is what Lynn talked about, are these moorings, where we have now passive acoustic recorders, and hopefully we'll soon get the data that spans the entire year and not just yeah, leaves out the summer, which was a very unfortunate technical glitch. And we want to connect these to the mooring data. In this case here, that's an example of krill acoustic backscatter observed by one of those moorings. And this is again very patchy, but what we can find is a really cool peak and increase in observed krill swarms in the spring and summer. But this is only one spot, and krill, yeah, as krill being that patchy and dynamic, we also want to have a look at this. Yeah, how does the spatial distribution change, and how stable are those hotspots? And one of the ideas has been to use an unmanned surface vehicle called the Sailboy that has been deployed in 2 years. Here's an example from 21. We tried to use this little wind and wave and solar powered vessel to survey transects north of the island for us, but ran into some technical problems. You can see there's a lot of closed dots here, and the whole thing didn't navigate the transect very well, but I mean almost useful. But then here it hit an iceberg and had to be brought out, picked up again for some weeks and repaired, and then tried to repeat it again. But it proved very tricky because this the navigational capabilities of this one is not that useful for actual transects. Like all of this, I think was like 3 weeks or so. So it's not a very synoptic survey then. But there's already something else in the works where hopefully next year we can try to use a new USV, which is called the Simrad Sounder, which has a whole galore of echo sounders and ADCPs installed to it, and it is motorized. It can go, I think, up to 10 or 15 knots, so it's really fast and can be either remotely steered or autonomously navigate along these transects. And that way, we really hope to get some really updated and much more high spatial and temporal resolution krill abundance data. And then we also want to use aerial drones to study to actually stop at one of those fin whale feeding aggregations and make some transects and get better counts and look at the abundance and the behavior in these aggregations. With that, I'd like to thank you. There's a lot more to be said, but I'll stop here.
Thank you very much for that one, Sebastian. I remind you all to pose any questions you have in the Q&A box. So for our next speaker, we have a pre-recorded talk. So I'll just ask the IT team to get ready to play Ayşegül Ergokça's talk for us, if possible. Thank you.
Hello, I am Reisinger. Today I'm going to present the detection of the food web structure of top predators in the Antarctic Peninsula by eDNA metabarcoding. Here is my presentation content. It is of great importance for peninsula ecosystem to determine the effect of global warming, climate change, and melting glaciers on peninsula biodiversity and the food web of species. In this study, it was aimed to determine the target species and biodiversity of the sampled habitats at the species level in the reference database scale with the environmental DNA metabarcoding method. Our target species are gentle chinstrap Adelie penguin, brown squid, beetle, elephant seal, and Antarctic shark. What is eDNA metabarcoding? DNA barcoding is molecular identification method. Metabarcoding is also molecular identification method, but you can detect more than one species. eDNA metabarcoding is samples come from environment, and you can detect sampled habitats biodiversity by using eDNA metabarcoding method. Sampling was carried out 9 different stations along Antarctic Peninsula. During second and third Turkish Antarctic expedition, fecal samples of Antarctic Peninsula top predator species were sampled for that analysis. Total 339 stool samples DNA extraction were performed with optimized DNA extraction method. One-step PCR amplification was performed with universal primers that amplify short regions of 18S eukaryote, CO1 for invertebrate, and 12S for vertebrate genes. Samples were added to the libraries at equal concentration by adapter ligation method, and high throughput sequencing performed using paired end amplicons of 300 base pairs long, and final approximately 1.5 million nucleotide sequencing per library were obtained after sequencing. Total, we had 9 libraries. For bioinformatics part, we use Obuchul's pipeline. Here is Obuchul's pipeline workflow. After various filtration steps for our data, we got 1 million filtrated trees, and the results filtrated sequence for matched with EMBL and in CBI database for eukaryotic vertebrate and invertebrate species. Here is our results. I have lots of charts, and I will briefly mention about my species results here. And for elephant seals, 2 eukaryote, 4 invertebrate, and 5 vertebrate species we detect related to the Antarctic Peninsula here. And for eukaryote, Pyrus antarcticus and Glommonas nivalis snow algae species we detect. And for invertebrate primers, we detect elephant seals with this primer, and we detect Euphausias parba, krill species, Cryptococcus antarcticus is springtail species, and Acunthus antarcticus is targeted species we detect here. And for vertebrate species, important species here is Notathenia cordyceps, which is fish species specific for Antarctic seas. And for Adelie penguin, 3 eukaryote and 12 invertebrate and 6 vertebrate species we detect related to Antarctic Peninsula here. Most common species we detect here, Anthelostica sigmoidea, which is Antarctic slab species, and we detect also snow algae here and diatom species. For invertebrate species, here we also detect most common Apus antarcticus and Cryodocra antarcticus, which is ice fish for Antarctic Peninsula, and also we detect here Antarctic springtail, and also we detect here the different krill species, and we detect Palmaria decipiens, and this is the marine red algae for Antarctic Peninsula. And for vertebrate species here, we detect also 3 fish species here. For delta penguin, we detect 20 eukaryote primers, 45 invertebrate and 13 vertebrate primers. And most common results along the species we found here, Desmococcus species and Epelota plana is krill parasite. We found that in Gento penguin dataset in here differently. Also, we found Hemicloris antarctica, which is also stone species and specific for Antarctica Peninsula. Here also we detect some snow algae species here. And for invertebrate primers, we detect here also the tardigrade species, springtail species, and the tree krill species here. And in different here we detect here Gondagenia antarctica, which is fish species, and we differently detect here also Neopagetopsis Iona, which is also ice fish here. And for the fish species, we detect here more fish species than the other dataset. 14 strap penguin, we 8 eukaryote, 12 invertebrate, and 9 vertebrate species we detect here. Similarly, most common species are here Desmococcus species and other snow algae species, but differently here we detect Phyresus antarcticus, which is Antarctic brown algae, and the Pseudoplerocoris antarctica, which is Antarctic yellow-green algae species. For invertebrate primers, also we detect here the tardigrade species and 3 other Antarctic krill species, springtail species here we detect, and we detect here Antarctic toothfish and Electrona antarctica fish, and here in invertebrate dataset. And for vertebrate species, we detect the fish species here important for chinstep penguin dataset. For brown squid, we detect here 15 eukaryotic species, and we detect similar results, mostly comparing the other species dataset. And differently here, we found 2 seal parasite species here. And for invertebrate primers, we also detect here the Antarctic krill species. And for the vertebrate results here, we detect here also fish species for important brown squid diet. And for Weddell seal, we found 5 eukaryotes, 7 invertebrate, and 6 vertebrate prime results for related to Antarctic Peninsula ecosystem. And for eukaryote primaries, we also detect similarly ciliate, small algae, and Antarctic toothfish here. And for invertebrate primer, we detect 3 krill species here also. And for vertebrate species, we detect 2 fish species also here. For Antarctic shark, 1 eukaryote, 6 invertebrate, and 4 vertebrate species we found for Antarctic ecosystem. And then the one result is Pyrus antarcticus, and this is algae species, and we found invertebrate species here, also fish species, and 2 krill species here for invertebrate primers. 1 fish species match with this data set. For discussion part, species detected in data set and not included in the peninsula ecosystem were excluded from our results. And samples were successfully determined as expected here. And Genta penguin was obtained as a result of matching almost every samples. And Eusopia sparba, Eusopia pacifica, and Tizonessa macura, those are krill species most common in Antarctica. And Nototania coricaps and Pedogramma antarctica fish species, we detect successfully in almost every dataset for 7 species, and also target organisms such as green freshwater, marine small, and macroalgae species specific to Antarctic Peninsula have been identified. On the other hand, in our dataset, match results were obtained with non-specific species for Antarctic islands such as soil, marine, freshwater, polar algae species, flagellates, layout, dinoflagellate lobster, crab, and the results are considered to be most closely matched species in the reference database for peninsula habitat. The matching results for Genta penguin were obtained as a result of matching with sardines and North Pacific herring, unlike other species dataset. Scientific research in Antarctica is significant to the future of global ecosystem. Thank you for your patience.
Thanks very much to the IP team for playing the recording for us. I'm going to invite our next speaker, Anand Pandey.
Thanks, Ryan. Namaste. And I'm Anand. I'm going to present my work. Actually, it's a big team's work which I'm going to present. Just a bit. Let me show you screen. Yeah. Okay. So I'm Anand, and I work at the Wildlife Conservation Society India, and previously I was working with the Wildlife Institute of India, where most of this work was done, and I'm also collaborating with multiple organizations in India as well as some organizations out of the country. The work is basically, what I'm going to talk about today is basically our ecological monitoring work, which we are doing under the Indian Antarctic program. This work was done for a period of, we started our work way back in the mid-90s, but there were very few participations, very few programs that we did that time. But later on, after 2013, we started participating in the Indian Antarctic program regularly, and we expanded upon our scope of work. So the first thing that we looked at was basically looking at seabirds and marine mammals in the southern Indian Ocean and Antarctica. And our focus was always East Antarctica due to the presence of our stations there. So giving a brief background, already I should not talk much about it, but you all know about the IPCC reports and the warnings that they're giving, which give a very grim future for the planet and also for the continent of Antarctica. And so in the last 6th assessment report, there are predictions that that Antarctic, the ecosystem, the food webs in the terrestrial as well as the oceanic ecosystems are changing, and which is happening very rapidly now. And this might also lead to a collapse of the krill populations in a few decades later on, and it might be catastrophic for dependent marine mammals and seabirds, although that is something which is the purview of long-term monitoring programs and long-term ecological research, which is being done under the purview of SCAR. For India, we have a national action plan on climate change, and we are also parties to the conventions on biological diversity and also the convention for the conservation of marine living resources. And we are not party to Convention on Conservation of Antarctic Seals and the Agreement on the Conservation of Albatross and Petrels, but it is also our national responsibility to feed information into it. We work at two sites in Antarctica. Basically, we have our presence in the East Antarctica, basically at LaRue Hills, which is here in the Prince Bay, and also in the Schumacher Oasis, which is in the Central Drowning Mudland. The two stations we have in Antarctica. Based on this background, we started our work way back in 2013, where our objectives were to look at monitor seabirds and marine mammals in Southern Indian Ocean and Southern Ocean, and basically with short-term goals of identifying distribution hotspots, range shifts, and abundance, and with the long-term goal, of course, with respect to the climate change. We are also looking at population demography and genetic connectivity in seabird populations, which breed in these two areas, which I showed earlier, Schumacher Oasis and LaRue Hills. Also, we are looking at the distribution patterns and abundance trends in ice breeding seals. Our aim is to implement a long-term ecological monitoring program and for wildlife in Antarctica. That's our goal. And with this information, we have basically started off with our long-term monitoring program. We are doing at-sea surveys. If you look at this sector, basically the Southern Indian Ocean sector, we start from South Africa to Antarctica, and this is an unequal site angle triangle, which we are basically sampling and trying to get as much information on seabirds and marine mammals in this particular sector. But our focus is also along the Antarctic coast, where between the stations, between the Schumacher Oasis and LaRue Hills, that is Prince Bay and Lazarus Sea, we are basically looking at how seabird and marine mammal distributions are. We have looked at different species. We have been able to record around 54 pelagic seabird species in this particular sector, and we are now closely looking at how these seabirds are distributed in this particular area. We are looking at particularly species which are of conservation concerns, basically large albatrosses and also some species of petrels and other species of shearwaters. We are also recording marine mammals in this area, so we've been able to record 16 marine mammal species with mostly dominating humpback whales. We are also documenting seals in the Antarctic sector. Basically, we have been able to document about 5 species of seals in our sector, and interestingly, we've got some sightings of Ross seal as well. And we've got Antarctic fur seal further north of Prince Bay. Now we're looking at, now our aim is to look at fine scale abundance distribution and habitat data for these seals, because now we know where to conduct our surveys and also post-processing of area data. For our seabird surveys, we focused our efforts in LaRue Hills, and we surveyed areas, and it was published recently. We looked at which species of seabirds are breeding there. We found 5 resident species. Emperor penguin breed a little east of Larsen Hills. They breed in Amanda Bay, but there are other species like snow petrels, Wilson storm petrel, and South Pole skua, which are breeding there. And apart from that, other species regularly pass through the area, and we record them. Our aim is to look at these 4 species now, looking very closely. Apart from Adélie penguin, which was not molting at Larsen, but it was molting a little east of Larsen on the Steins group of islands. But their numbers, like we are still confused about their numbers, but we are doing this work to understand how the species are distributed. Like snow petrels, there are 4 major colonies which we know now, and we have initiated this work to understand what is the nest density, and it's about 6 nests per meter square. For especially for South Polar skua, there have been around 21 pairs which are operating in this particular region, and Wilson storm petrel is there but sporadically nesting. For Adélie penguin, there's a widespread molting areas, and if you look at towards the east of this particular area, this is the island group of Larsen Hills, and east of this is the group of Steines Islands where Adélie penguins are molting. And if you look at this particular image pattern, we are looking at how Adélie penguin population is changing in this area, and we are using particle analyzer tool in ImageJ to understand how many breeding pairs are there, and we've got about an estimate of about 800 odd individuals over there, but this needs to be repeated because this was done way back in 2016, and in 2019 we could not repeat most of it. Then we are also doing motion. We are also recording the breeding behavior of snow petrels in the area. We collaborated with IIIT Delhi, which is an organization based in Delhi, and we developed these motion sensing cameras. We designed them and put them in snow petrel nests to look at how the breeding is happening in snow petrels, and basically we've been able to record the sequence of breeding in the area, but also looking at now to automate this and to fix kind of solar panels to understand how these to continue this kind of monitoring, and to understand if we can put more cameras across. These were just 5 pair of cameras that we put, but we are planning to expand this and put more cameras on the ground and look at how the breeding is changing or patterns are changing over a period of time. So this is just 2 years of data that we have, but of course, this doesn't say much about what's happening to the snow petrel populations, because the last set of data that comes in shows a lot of mortality, but we are not really sure of what's happening. So we need to go back and do the work again. We are also, for the Schumacher oasis, there's not much, there are not many species of birds breeding there, but there are the South Polar skua, which are nesting in the area, around 7 pairs of South Polar skua which are breeding there. No other species of seabird breeding there, no snow petrel we recorded during last 4-5 years of monitoring, and also only stray events of Adélie penguin breeding in that area. And this is just a couple of events which happened over there, but most of the sightings that we had was the vagrant sightings. We also undertaken a genetic monitoring of 2 species, basically Wilson storm petrel and snow petrel, and we are basically looking at how the phylogeography is there for snow petrels. These are the 3 sites actually we collaborated with Dr. Anna Terrell, and we got some samples for Swartmara Hills. Now we have samples from these 3 sites, and we are looking at how the phylogeography of snow petrels is in this area, and using the cytochrome B gene. By sequencing the cytochrome B gene, and we are looking at the haplotype sharing, so a lot of mixing. And there was a recent paper by Carey et al, and they said about high vagility, that is high movement between snow petrel populations across Antarctica, and they were basically looking at the Australian stations. But for our data as well, we are seeing similar kind of patterns. We have also undertaken geotagging of South Polar skua, put some geolocators on South Polar skua to look at their movement patterns. Unfortunately, this happened just before the COVID-19 pandemic hit, and we could fit these geolocators, but we have not been able to recover them so far. We are also looking at cetacean distribution in the Southern Ocean, the same sector that we are operating, and also preparing a manuscript on the distribution patterns of cetaceans in this particular sector. to look at the way forward, we are looking forward for collaborations on various fronts. So right now we are focusing our efforts on seabirds, specifically snow petrel and the Wilson storm petrel and the South Pole skua. These three species of birds we are focusing our effort. We are also willing to work further on seals. If anyone is interested, please email me. My email is given here. And we are basically looking for stronger collaborations to strengthen the Antarctic wildlife monitoring program, which has been hit after the COVID-19 pandemic. Actually, I would like to thank the ministries of Environment, Forest, and Climate Change and Earth Sciences in the country in India, and also my team members of various expeditions. This information that I presented here belongs to 5 expeditions, and my team members basically Shashank and Ankit, Yogesh and Dr. Sani for helping us out in this conducting this monitoring program. Thank you very much for your kind attention.
Thank you very much for that talk, and I'm perfectly on time. Just a further reminder to ask any of those questions in the Q&A box. And while you do that, I'd like to invite our final speaker for the session to present, and that's Nobuo Kokubon. In general, I apologize for my pronunciation of your names and surnames. Thanks, Nobuo. We can see your screen and cursor, but we can't hear you at the moment.
Can you hear me?
Yes, we can hear you now.
Ah, okay. Thank you. Sorry for
Perfect.
that. And thank you, Chairperson. In this presentation, I'd like to introduce our recent work on the Antarctic coastal oceanography and its influence on the foraging behavior revealed by the wetland seals. First, I would like to introduce the background of our work. Antarctic coastal marine ecosystem is characterized by the extensive seasonal sea ice and land-fast ice and consequent high biological production. In recent years, an abrupt changes in the sea ice extent is reported, and its potential influence on the local marine ecosystem attracts attention. Thus, there is an increasing demand for deeper understanding of oceanographic processes and temporal changes in the coastal areas. From this context, deep diving marine mammals have been used as a platform for oceanographic surveys. For example, southern elephant seals, one of the deepest diver up to 2,100 meters and migrate over long distances, equipped with the miniaturized oceanographic instruments, revealed oceanographic conditions of oceanographic structure across the Southern Ocean. Nevertheless, oceanographic structures on the continental shelf covered by land-fast ice are still not well known or covered by these animals. Japanese Showa Station, East Antarctica, locates such environments with extensive seasonal ice and land-fast ice with considerable seasonal and interannual variation compared with other locations. In such environment, wetland seals inhabit commonly. Thus, this study aimed to reveal oceanographic conditions in coastal areas covered by land-fast ice during autumn and winter by the instrument wetland seals, and to explore the ocean circulation and its influence on the local marine ecosystem. We deployed the miniaturized oceanographic instruments called CTD tags on 8 weather seals near Showa Station in 2017, recording temperature, salinity, and diving behavior, and obtained enough data from 7 seals. Then, water mass and its influences on foraging behavior was analyzed. These are the sea ice distribution, wind force and direction, and the sail track. In April, sea ice broke up extensively, and at the same timing, easterly wind, shown by these black arrows, became strongest. And from March to June, most sails moved toward east, about 80 to 600 kilometers along the continental shelf. And the figure shows the temperature and salinity data obtained from the sails during autumn and winter. We plotted all the data like this and classified all the data into these 5 water types using some definitions according to previous physical oceanographic studies. These are black winter water, Antarctic surface water, modified circumpolar deep water, modified shelf water, and supercooled water. And this figure shows the time series of the vertical distribution of the 5 water types. These 2 panels show the examples from the 2 individuals. Through the examples, the winter water, the black dot, low temperature and salinity water, was most prevalent in the area through autumn and winter. A point is that warm water such as the orange Antarctic surface water in the subsurface appeared in the subsurface, and the depths became deeper as the time progressed, and the red modified circumpolar deep water appeared in the deeper depths in autumn. Also, the blue supercooled water appeared in the surface after May. And these figures show the horizontal distribution of the 5 water types. These rectangles show the expanded part of the maps. The low temperature and salinity water, the winter water, was most prevalent in the area through the time. Here again, the orange Antarctic surface water appeared widely on the shelf in April, whereas the red modified circumpolar deep water appeared in particular areas like these bays, and the blue supercooled water appeared widely after May. During autumn, waters on the shelf may be cooled by the cooling weather conditions. So why warm Antarctic surface water appeared on the shelf during autumn under the cooling conditions? We consider that one possibility is that warm water was intruded from the off-shelf areas by wind stress during autumn. These panels show the month-to-month wind force and direction along the coast, where the blue color indicate easterly wind and red color indicate westerly wind. If land-fast ice breakup and strong easterly wind blows like in April, it enhances shoreward transport called Ekman transport of the off-shelf warm surface water. As a result, near the coast, the surface water is pushed down to the deeper depths and cooled from the surface. Such process is called Ekman convergence or downwelling. These panels show the estimated depth of the Ekman downwelling month to month, and the depth became deepest in April, as the actual data showed. Furthermore, warm water had positive influences on the foraging behavior of the cells. This figure shows the effect of different water types on the index of foraging behavior, where positive values indicate positive effects and negative values indicate negative effects on the foraging behavior. The orange Antarctic surface water, red modified circumpolar deep water, and black winter water under Antarctic surface water had positive effects on seals foraging behavior. So why these water types had positive effects? We consider that the additional prey was transported by the water intrusion. This is the cross-section schematics of the continental shelf during the study. Surface of the continental slope or off-shelf areas are known to be the main habitat of Antarctic quail. Also, epipelagic layer of the off-shelf areas are known to be the habitat of mixed-feet fishes. During autumn, water intrusions from the off-shelf areas may enhance prey transport as well as foraging, seals foraging behavior, as the figure shows. For the summary, wettle seals from Showa Station showed long distance and 600 kilometers migration towards east along the coast during autumn and winter. And warm water such as Antarctic surface water and modified circumpolar deep water was observed in subsurface and deeper depths where the seals visited. And water intrusion from the off-shelf areas may enhance prey transport as well as seals foraging behavior during autumn. This paper was published in Limnology and Oceanographic in last year. And that's all. Thank you for listening.
Thank you very much for that interesting presentation, Nobuo. That was the last talk for our session. So I'd like to thank all the speakers for presenting and to you all for attending and listening. We do have around 15 minutes left for general discussion and any questions you'd like to put to the speakers. You can either answer, beg your pardon, ask those in the Q&A box, or you can, I think, raise your hand and we can enable you to ask those questions directly. So I'll just give you a few minutes if anybody has anything they'd like to ask our speakers. Yes, Michelle.
Thank you so much for that talk, Nobuo. That was really interesting. Just one quick thing I noticed. I noticed the 900-meter depth that you showed kind of at the beginning as a maximum depth. Is that a new record for Weddell seals? Is that like a new record maximum dive?
Thank you for your question. Yeah, I saw the record in other paper, so it's from not our data, but yeah, it's from other area in the Antarctic, I think.
Oh, I see. Okay, thank you.
Yep, thank you very much.
Thank you, Nobuo. Lynn Asverstadt also just asked me to say that she had to leave the session, but she's happy to take any questions by email. Yes, Anand.
Oh, sorry. This is my mistake. I'm sorry.
No problem at all. Horst, please go ahead. Let me just. Okay, there we go. You should be able to speak now, Horst.
Thank you. Thanks, Nobuo, for this inspiring talk. I do have a question that also combines your CTD tech deployments with your investigations in seal cameras. When you do have warmer waters arriving on the shelf, for example, also due to undulating frontal systems, you would have a melting effect on the underside of the ice shelf. And this you would actually observe in the structure, in the surface of the ice shelf. Did you make investigations with cameras at the same time that potentially showed the underside of the ice shelf?
Okay, thank you, Horst, your question. So we tried to get some underwater video images, but it was not successful during winter. But yeah, actually, we deployed some videos after a few months in spring, and we obtained some clear images of underwater foraging behavior of seals, especially underneath fast ice, and we obtained some video images of Antarctic krill feeding by wetter seals. Yeah, these are examples. So I considered that some water intrusion from off-shelf areas may enhance such biological interactions between sea ice and ice algae and Antarctic krill. Then foraging condition was enhanced, but we need to confirm this by attaching video during autumn and winter if we could, I think. Is it the answer for your question?
Yes, this helps a lot, Nobuo. Thank you very much. And the images you just showed are very inspiring if I see this huge accumulation of krill that potentially is available for the battle seals, let's say, as a bit of junk food underneath the ice. This should throw a new light a bit on the foraging ecology of the battle seal that we consider always the fish dependent. And if we see these huge amount of crustaceans, I would say that might be at least in areas like Shoba Station to be revisited. Thank you very much.
Thank you very much.
My question is for Sarah, if that's okay. Sarah, I was wondering, could you give just real quickly what some of the primary characteristics of the different groupings were? So like that little grouping of colonies kind of in the just that like grouping of like 3 colonies, what were the primary like habitat characteristics for them versus like the wet LC? And do you have any insights as to whether or not those matched the genetic metapopulations?
I thank you for your question. I don't know about the genetic population. I haven't looked at it yet, but I can share for the characteristic. I can share my screen and show you one thing. Do you see my presentation?
Yes, that's perfect. Thank you.
Okay, so the 3 dots here, we are talking about that, right?
I think so. I lost your cursor, but yes, that's right.
Okay, so it's the blue here. And it's interesting because it's characterized by relatively high fast-size persistence, 75% persistent compared to other cluster, and also high fast-size extent. So probably a small pocket of an area where you have like an important range of fast-size, but negative trends over time, and intermediate magnitude, so not too low, not too strong. So it's probably a special fast-size area. I mean, like a little bit more persistent and further extent than others colony. I don't have the plots here with the biological factor, but I can send it to you and we can discuss. But I don't know. I will look at the genetic linkage and see if they are very. I mean, why they are there, and why it's different like that.
No, that's really great. Thanks. No, that's interesting that the fast ice extent, I assume fast ice extent was higher in those areas. It's interesting to me that those colonies are small but persistent, even with like greater fast ice extent and greater fast ice persistence. So thank you.
Yeah. Thank you. I mean, just to complement one thing, it's interesting. I mean, what I was surprised about the study is the difference. So the difference in the habitats, if you look at really variable by variables, between different clusters, you have very different habitats. So it seems like the emperor penguin can be in very different type of fast-ice dynamic and stuff. So it's a good thing, but I mean, we still need fast ice, of course, but it's a good thing in terms of how they can adapt to different environments.
Great, thank you.
Thank you, Sarah and Michelle. Monica, do you have a question?
Yes, good. Hi, good, as you said, Ryan, good for wherever you are. First, just a comment. I want to share my experience that I didn't know that if I raised my hand, I had the option of asking a live question. So maybe for the next sessions, people could be told that, because I was wondering how I could ask a question without going through the Q&A. But anyway, the question goes again for Sarah, and if you could share again your presentation where you have the areas. It's the same kind of figure, but the one with the kind of like the pie where you have the different areas. If you could just explain a little bit to me that division. Is just that I see different colors in they don't seem to be exclusive. I see some red dots where oh wow. But anyway.
Do you see my presentation now?
No, what I see is your R script page, I think. That's why I said that was
very
not
impressive.
but it was cool. Thank you.
Working on it. Okay, this one, sorry. Do you see it? No?
Yeah, I think it's number 17. If you go to number 17, where you have slide 17, where you have you have the X there, I quite didn't understand. There are some blacks and blue, reds on the West Antarctic area, and apparently that's not where these dots were supposed to be. So could you just run over the classes and how you got that division as you did? Thank you.
Yes. So we did a model based clustering on the principal component analysis. So you take 2 dimensions, 3 dimension, 4 dimension. We took 4 dimension and we ran a model based clustering that give us 5 different classes that are here. And as you can see, I randomly put these lines because I was trying to interpret the different classification between the region. And we can see that we have a region here dominated by the class 1.
Okay.
Is that okay? And then we have a second region where we have a domination of the kind of class 5, but we still have other colonies that are not class 5. And especially it's interesting this pocket of 3 colonies that is the only colonies. They are the only colonies where you have the class 3 around Antarctica. So it's a very special environment, and it's what we were discussing with Michelle. And then you have this other region where you have like mostly the class 2, but you still have class 4 in the middle. And then here you have kind of a mix of everything, which is also interesting. It's also based on 13 variables only, and the 4 dimension of the principal component analysis, taking into account about more than 60% of the variability. So it's what is the habitat given these variables and the principal component analysis. So it's a way to characterize per colony what kind of habitat and what are the habitats that are similar.
No, that's great. It's just what I missed was that you just said now mostly these are the areas where you mostly find classes such and such, and that's all. Anyway, thank you very much.
Thank you for your question.
Thank you both for that discussion. That brings us to the end of our session, and I'd like to thank all our speakers again for a series of stimulating talks, as well as all the attendees for, sorry, just checking the chat, all the attendees for attending, and for your questions to the speakers. Our next session will be tonight, I believe, at 9 p.m. UTC, and looking forward to seeing many of you there, as well as new faces for whom it's a more convenient time zone. Please enjoy the rest of your day, evening, morning, wherever you may be. Thank you.
Thank you.
Thank you.
Thank you very much. Bye.
Thank you. Bye.
Thank you, IT team, for your help. I appreciate it very much. It all went smoothly. Thank you very much.